databases. So what are the major aspects of bioinformatics? So we discussed about various aspects. So what are the major aspects of bioinformatics? Oh, that's good. Yeah, NPTEL lecture you attend, then you have to answer all the questions. Yeah. Only database. And again, big data, tools, hypothesis, drug design, and so on, right? So then we discussed about various databases. What are the DNA sequence databases we discussed yesterday? DDBJ, EMBL, yes, GenBank, right. So literature, we discussed various literature databases. What is the literature database specifically for biologists? PubMed, right? So then we discussed about other databases which are common to all fields of science and engineering, right? So the other database we discussed, which is common to all disciplines, Scopus, Google Scholar, and so on, right? Okay. Then we discussed about the protein sequence database. What is the universally available protein sequence database? UniProt, right? Fine. Then we use a tool, right, that we developed in our lab to derive some features from the sequences. So what is the tool we discussed yesterday to get the features from okay, fine, the sequence feature, okay. So now today I'll discuss about some of the features, how to get the features, not just the from the program, right? And what are the various aspects we can do with these protein sequences. Okay, so I'll share the screen. Right. Okay, my screen is visible, right? Okay, today we discussed about the protein sequence analysis, right? So what can we do with the protein sequence analysis? Okay, so what is the building block of proteins? Amino acids, right? So then if you have how many naturally occurring amino acids? 20 amino acids. So these 20 amino acids, right? So these are all the specific 20 different characteristic features. They are polar, non-polar, charged, and so on. Then from these amino acids, how to form a protein? Right? So I'll tell you, okay, so if you see this is an amino acid, okay, this is the structure of amino acids. Here is the amino group here, carbon group here, and we have hydrogen here, and the R1, this is your side chain. So here this is the amino acid 2. So they form, they are combined together by the elimination of water molecule, right? This OH and the guys. So elimination of water molecule, they form a bond. Okay, this bond is called peptide bond, right? Peptide bond formation. So if you combine 10 amino acids, how many water molecules will be eliminated? 9. Okay, because it is for the n minus 1, because it is 2 minus 1, 1. So if 10 means 10 minus 1 equal 9. Fine. Okay, how to form this sequence, right? So now there are various amino acids, they join together, right? So is it possible to combine any amino acids as you like to form a functional protein? There are 20 amino acid residues, right? So if I give you 20 residues and give you the length of 100 residues in a protein, you can make plenty of combinations, right? So if you make any combination, so can you get the functional protein? Right? No. So then how to define the functional protein? So I'll tell you. Okay, here this is the 20 different amino acids. You can see the beads here, and here this is the main chain. So here this is always called, this is the same. The main chain is the same, right? But the beads are different. The beads I put the different amino acids here. So you can change the beads as you like, right, to form the peptides or polypeptides, right? Okay, like you have the 26 alphabets in English, so you can have several articles, books, and many things, letters, parts, everything you can form. Likewise, use this 20 amino acids, right? You can form the proteins. But if you have this letter, for example, if you write like this, you can see this word in the dictionary. But if you use this one, university, you can also see the words in the dictionary. But if you write like this, automatically your word will tell you the error because these words are not available in the dictionary. Likewise, you can make any combination, but these combinations cannot form a functional protein. So nature selects a specific combination of amino acids. So it knows, okay, this is a specific combination of amino acids, right, to form a protein for its function, right? If you make any changes, if you disturb any changes, right, then it will have different adverse effects. For example, sickle cell anemia. So here, if you change the glutamic acid to valine, right, so here it forms a hydrophobic environment, so it makes fibrils, and so it has this blocks this getting this oxygen, and so you have these breathing issues, right? So if you have any specific mutation, change any of this order or change any of the amino acids, you will have the effects. There are a lot of diseases mainly because of these mutations. So nature selects a specific combination of these amino acids, right, for a functional protein. Fine. So this is an example. This is a hemoglobin, right? You can see this is a sequence because if you this is valine, this is the histidine. These two residues join together by the elimination of water molecule. Likewise, so join together to form the functional protein. If you see the hemoglobin, you can 6, 3, 4, 5, 6 is a glutamic acid. So if you convert to valine, so it's what is causing the sickle cell anemia. Likewise, yesterday I showed some of the mutation databases developed in our lab. And if you look into the databases, you can see various proteins and various mutations and how these mutations which change the function or change the affinity or cause disease or not. Like other databases called Cosmic, other databases available in literature. So the Cosmic is mainly for the cancer mutations. If you look into that database, you can identify the different mutations in patient samples, right, which can cause cancer. Fine. So now this is the primary sequence, right? Because this is the sequence of amino acid residues, and this structure is called the primary structure because there is a starting structure which contains only the amino acid residue information, and this is called the primary structure of a protein, right? So there are 4 levels of protein structures, right? Primary structure, secondary structure, tertiary structure, and the quaternary structure, right? Okay, how to define these type of structures? How does the different organization of these proteins? Okay, primary structure, it tells you just the sequence. Okay, so what is M? M is for methionine, right? This linear arrangement of amino acids. Right? Then these amino acids, they form some specific patterns. Right? They have some regular structure in the structure. That is called secondary structure. So likewise, I think sometimes this is like a spirals. These are also called helices because this is first formed in the and they first observed in proteins. So they put the Greek letter alpha. So here the name is called the alpha helices. Likewise, some of them they have the ladder type of structures. Okay, in tomorrow's class I will explain more about these things. So you can see some of the dots here. These are called hydrogen bonds. So they form the hydrogen bonds to form this ladder type of structure. This is the one observed second time. So this is why they mentioned the beta because the Greek letters alpha, beta, gamma. So it's alpha helices, and this is called the beta strands. Right? Now this is a regular arrangement, the periodic arrangement of this primary structure, this amino acid residues. So that is called the secondary structure. The tertiary structure, it forms a global shape. So in this case, the atomic coordinates of each atom are exactly known. We are using several experimental techniques such as X-ray crystallography or NMR spectroscopy, right? So you can exactly tell the location of each atoms in the protein or in this amino acid residue. So here completely we can know that, right? So that is called tertiary structure. Tertiary structure contains the primary sequence information because it has the regular secondary structure information, as well as it can have the information regarding the atomic level coordinates, right, for each atom. You see here, these are all the alpha helices, right? This look similar. You see here, this and this look similar, so they are the beta strands. Then the quaternary structure, so the assembly of these subunits. So here there is only one. Sometimes they 2 or 3 join together and form a complete structure. That's called the quaternary structure. Here either they can be similar, same, or the chains will be different. If it is same, they are called the homo. If it is different, it is called hetero. Right? Depending upon the dimer or trimer or tetramer, depending upon the number of subunits in this quaternary structure. Right? So what are the different levels of protein structures? Okay, what are the different levels? Yes, four levels. What are the different 4 levels? Primary, secondary, tertiary, and quaternary. What is the information you get from the primary structure? Primary structures will tell you only the amino acid sequence, right? There's only the sequence, you know, the arrangement of these residues. So what are the information we get from the secondary structure? Okay, so this is periodic arrangement of these amino acid residues. There are different levels, like this is the different secondary structures, like the alpha helices and beta strands. Fine. So what is the tertiary structure? Which information we get in tertiary structure? Atomic detail. So you get the coordinates of each atom, right? Fine. So in the quaternary structure, right, the subunits, the combination of subunits, the assembly of subunits will give the quaternary structure. Fine. Okay. So now we will see today's class mainly focus on the primary structure. Okay. Tomorrow's class I deal with the secondary structures, and the day after tomorrow I will talk about tertiary structure. Okay. So okay, what can we do? For example, if you have the one sequence like this, right? What are the various information you can obtain from this sequence? And why this sequence is so important? Right? Okay, so we can derive various features. Yesterday we discussed some of the features in the practical lesson, right? But just we showed how to get the features. But today I will explain how to get the features and why the features are important. Right? The easiest one, the simplest one, that is called amino acid occurrence. Amino acid occurrence will tell you how many times each amino acid occurs in a sequence. For example, if we have this sequence, right? So how many times A occurs in this sequence? Only one, right? How many times C occurs in the sequence? 0, right? How many times S? How many S in this sequence? 2, right? So this is just occurrence. This will tell you how many times each amino acid occurs in the sequence. Okay, now this is just an example I put. Okay, actually I put this is a peptide. This is what I wrote here. Okay, so here I give a sequence. This is exactly the same sequence. This is the protein sequence, right? This is a real sequence. So if you see this one, right, what is the occurrence of alanine? So if you see, we can count this because now you can see 15 alanine here. So the occurrence is 15, right? If it is C, it is 2. Right, likewise you can see 20 different amino acids here, right, and here the occurrence here. So what is the occurrence of F from this sequence? 8. What is the occurrence of tryptophan? 2, right? Yeah. So how you got 7? Okay, because this tryptophan you need to know the single letter code. The single letter code for tryptophan is W, right? So W is here. So this way it is 2, right? The answer is 2. So you can see the occurrence of tryptophan, right, in this sequence, it is 2. If you look at here, you can see 2 places, one here and one somewhere else. Where is it? One is here. Right? So you can see 2 tryptophans in this sequence. So the occurrence is 2, right? Fine. The simplest one. So here, so one aspect we can give the numbers. So if one protein has 100 residues and another protein has 1000 residues, right? If you see the occurrence, almost all the residues in the 100 residue protein are less, But compared with the 1000 ones, okay, everything is at least in the 10 times more, right, than the occurrence which you observed only in the case of the small protein. Then how to see what the preference of very specific residues, right? In this case, we need to normalize, right? So I define another term that is called composition. And the composition we define as n. It's not n of I. This is n. Okay, for example, it's I, then it is n of I. Multiply 100 divided by n. There's no summation here. Okay. So for any residue I, right? So get the occurrence. Occurrence is the number of times each residue occurs in the sequence, right? Multiply 100 and divided by n. Where I stands for any residues, so n is the number of residues. Okay. So what is the composition of alanine? So totally how many alanine in the sequence? 15 alanine. Multiply 100 and total number of residues 147. So it is composition is 10.2%. Right? Now what is the composition of phenylalanine? What is the composition of F? Uh, 8 divided by 8 divided by 147. So what is the composition of tryptophan? Uh, 200 by 147, right? Right. So you can find. So you can W2, so 100 by 47. So you can see that now you can normalize it, and now you can tell which residues the more occurrence, or what is the composition of each amino acid residue, right, in a particular protein. Okay, so we can write an algorithm. So how to computationally calculate for one protein? You can easily do it. If you have thousands of proteins, right? How many protein sequences in UniProt database? How many sequences in UniProt database? Anybody remembers? 30 million? Any other numbers? 88 million. Say any other numbers? Oh, you got the good correct number. Yes, 189 million sequences in the UniProt database. So yesterday we saw the rarely occurring amino acids. What are rarely occurring amino acids in UniProt database? Tyrosine, not exact tyrosine. Okay, close to that. So two others. Tyrosine is relatively less. What are other two rarely occurring amino acids? Very less percentage. Sulfur is not an amino acid. Sulfur containing amino acids. So you're right. Yeah, cysteine and yeah, methionine you can say that will be more, but less one is the tryptophan, right? Tryptophan, cysteine you can see the rarely occurring amino acids, right? Okay. So now if you have this many million sequences, right, you need to write a code to get the average, because these numbers they derive from the calculation of all the amino acid sequences in the UniProt database, right? So in this case, we need to write an algorithm. First, what are the different inputs do you need to write an algorithm? So what are the inputs we need to calculate the composition? Amino acid sequence, right, and 20 residues, right? So 20 residues we need, and the amino acid sequence we need. First, this is your sequence. Read the sequence. These are 20 residues. Okay, I equal 1 to 20 different residues. And here you read the sequence. You have the sequence. Now first normalize, because you have to put everything 0. Initialize the composition. Initialize occurrence, right? Each is 0. Otherwise, computer will take whatever they want, right? So in this case, you put 0, and then you take the sequence and check the first sequence with the 20 or 20 residues, and while it finds a match, then you put that number as that array as 1, right? Then go to the second one, search with all the 20 residues. If they don't find the match, repeat it. If they find the match, then where they find a match, you add 1, right? Then they repeat all the sequences. By here you can get the occurrence of the residue, right? All 20 numbers will get. Then you normalize it with the total number of residues, right? Then you will get the composition. So we need the sequence and the residue and match whether these matches or not, and where we find the match, so it will add the numbers, and finally count it for all the residues in the sequence and get the occurrence and normalize it so that you can get the composition. This is a sequence. I shown sequence here, right? So this is the 20 residues. Repeatedly you check the match, and finally these are the number of matches. So here you have this occurrence, and they normalized with this 79 is total number of residues in the sequence, right? So then you divide by 79, and here you get the occurrence, right? That is fine. Okay, so now the composition, right? Here we have the web resource, right? So this is PIR. PIR is the initially developed the database for protein sequences, and it has a lot of tools in this database. So go to search analysis here, and here at the end you can see the composition molecular weight, right? If you click on this composition or molecular weight, right? So then go here, it's asking for your sequence, right? You write your sequence here and then submit here, right? And then you'll get the composition here. So this is your sequence. This is what I showed in the first slide, right? And here this is your amino acid occurrence, right? And here this is your amino acid composition, right? So see, if you see, here it's a methionine and the cysteine and the tryptophan, they are also very less composition, right? The UniProt, they give the average of all the sequences. This is for this particular protein, right? Fine. So the next one is a molecular weight. So we discussed two aspects. One is occurrence, and the second one is a molecular weight. And now we talk about the second is the composition, and we talk about molecular weight. So molecular weight, right? It is given as a summation of n of I, where n of I is the number of times each residue occurs, right, in the sequence, multiplied by the weight of these amino acids. Okay, you can give the weight of all the 20 amino acids here, minus 18 into n minus 1. Why you have to do this minus 18 into n minus 1? What is the meaning of this 18? So molecular weight of water. Why n minus 1? Why do you want to multiply 18 multiplied by n minus 1? N is total number of residues, right? Then why n minus 1? Yeah, that is because if you have the n peptides, n residues, right? N residue peptide, that's release of n minus water molecules, right? This is your reason. Okay. So now can you calculate what is the molecular weight of the peptide AIKL? Can someone get the answer for this? AIKL. What is the molecular weight of A? Or single letter is here, A is this one, and I is isoleucine is here, K is lysine is here, leucine is here, and can you get the molecular weight of this peptide? Approximately? The number? $2,500? Is very high. Okay, so you need to get the n. This is this A of molecular weight is here, and then plus molecular weight of I plus molecular weight of K plus molecular weight of L minus 18 into n minus 1. Any other number?
Underwear.
What is the value for A?
Underwear.
89, fine. What is the value for I? Correct. What is the value for K? 1 case. What is the 140? Okay, and what is your L? L is 131, right? What is the value of what is the value of K? K is fine, 146. And what is the value for L? 131.17. So what is our total molecular weight? If you add all these four, what is the number? Four 97. Then we need to subtract how many water molecules? 3. So 3 multiplied by 18. What is 3 multiplied by 18? 54. So you subtract 54 from this 497. Correct. Okay. So you got 443. Okay. This is the answer for the this peptide. Okay. So any peptide, if you give, right, so they can calculate right for the molecular weight of each residue and subtracted 18 to minus 1. So for this sequence, okay, here is the data. Okay, here is the molecular weight 15998. That is a full sequence because they calculate for each residues minus 18 into 146. This is this number. Okay. So you can calculate, if I give any peptide, you can easily calculate the molecular weight, right? Fine, good. Okay, now the next one is the properties. Okay, so yesterday we discussed, that's correct, that's good. Yeah, so you can do the properties, the various properties. Can you list some of the properties of amino acids? Yesterday, the C2 picture also we showed a lot of properties. So we got the numbers for several properties. So for example, if you can take the 20 residues together. Okay, 20 residues together. What are the various aspects? What are different numbers you can derive? What are various properties? Okay, one is hydrophobicity. That means see whether it is hydrophobicity or polarity, and then color, yes. And we have the weight, and we have the shape, and we have the size, how the flexibility, and we have the propensity, correct? Propensity for helices, propensity for the strand, propensity for the coil, and we have the free energy, enthalpy, entropy, right? So there are various properties, right? We have the amino acids. Okay, here I show you just for the sake of explaining a property. The numbers, okay, just I made simplicity because I want you to do some calculations. That is the reason I put the simple numbers, right? So for example, take any property. For example, here hydrophobicity. Hydrophobicity is given as n of I, that is number of times the residue I occurs, multiplied by the hydrophobicity of that particular residue. So take the summation over I equal to 1 to 20, right? You will get a total value, right? Okay, so here I put ACGMY1, FILLBW2, DEHKR minus 2, and NPQST minus 1. Okay, so you have some reasons to put this. It's not just random. Can you see the numbers 2? Which type of residues these two? If you see the residues, can you distinguish the residues with numbers 2 and minus 2? they are from some specific group. So if they take a -2, they have some specific property. So can you tell what is specific property of the residues which I put for -2? Good. So polarity, that is very common. So can you more specific? If you see the glutamic acid and aspartic acid, glutamic acid, so they are from which category? Negative charge, right? Acidic, right? So if you have the HKR, how about the HKR? They are positive charge, right? Lysine, arginine, positive charge. So these are all charge residues. So this way I put everything together, charge residues together, because we are talking about hydrophobicity. So they all water like loving amino acids, right? That is the reason I put as -2, right? That is very favorable, right? So then I put the +2. Can you tell something about the residues which I give +2? Can you see the type of amino acids in the +2? Right. If you see, this is aliphatic, ILV or aliphatic residues. It's not polar. I am talking about the +2. And which belong to F and W? They belong to some category. What is the category F and W belongs to? Aromatic. They are aromatic. These are aliphatic. So I put -2. So these are all small aliphatic residues and other residues, sulfur-containing residues. I gave 1. These are the polar residues. I gave -1. Okay. So it looks like random, but it's not random. Okay. So only with some reasons I give these numbers. I give the numbers because of the calculation purpose. It is easy to calculate because you need to calculate the values here. Okay. Okay. So now I give this sequence. This is your value. This equation, and I get two sequences. Could you calculate the total hydrophobicity for sequences 1 and 2? Someone, could you please quickly calculate what is the value for sequence 1 and what is value for sequence 2? Total hydrophobicity. What is the value for A? 1, right? So you can do it, substitute this one, and what is the total value? Right, we give the values for all the residues. Finally, calculate the value for sequence 1 and the total value for the sequence 2. -4 for sequence 1. Anybody else got the same number? One, two, 0, -1, +1, 0, 1, -1, -2, -4. Okay, right. Okay, 2, 3 participants got the same number. So -4 and 0.4, right? What about the second sequence? $13. Anybody else got the same number? Okay, fine. So now if you see first one, it is -4 and the second one is +13, right? So what can you tell from these numbers? Can we tell something from these numbers? Because it is -4 and this is +13. What's the meaning of these numbers? So what can we tell from these numbers? Okay, so more positive means more. That means this residue is highly hydrophobic. If you see the second residue is completely hydrophobic residues, and first residue it has like lot of polar residues and charge residues, right? So this will tell you some of the characteristic features of proteins. For example, if you take the transmembrane helical proteins, that means the proteins, the segments are embedded into the membrane. Membrane environment is highly hydrophobic. So if you see the transformer regions of the alpha helices, so they are highly enriched with hydrophobic residues. So if you find a peptide like that, but at continuous stretch of 18 to 24 residues, we can tell that these residues mainly located in the membrane. So it could be probably a transmembrane protein, right? So that we can easily tell. If you get the hydrophobicity is very high, you can tell that this could be a transmembrane segment. Okay. On the other hand, if you have a charge, if there is highly polar residues and the positive charge residues, then you can tell that this could be probably a DNA binding domain because it has high tendency to interact with the DNA. So that could be the probably DNA binding protein, right? Right. So if you see some of the features, you can easily infer, you can classify these proteins right based on the functions. So several proteins are unannotated. So we don't know exactly the structure or function. So we can use these physicochemical properties to annotate these type of sequences. That is the reason why we need these type of properties. Okay, fine. So I put some numbers, right? 1, 2, minus and minus 2. I'll tell you exact numbers. This is okay. Okay, so that slide is missing. Okay, so actually exactly we have the numbers for that for the 20 residues, right? Either you can obtain from the experiments, or you can just one moment. I'll tell you. Easily. Anyway, it's fine. So we have the exact numbers for the 20 amino acid residues. Either they obtain from the thermodynamic transfer experiments, right? Like the relative solubility of water and ethanol or water and octanol, right? Or they can derive from the 3D structures. If you have the protein 3D structures, you can derive these values based on the location of other residues, right, which are in the neighboring of that particular residue. I will explain this in details in the next class when you talk about protein 3D structures. Okay, so now the question is, okay, so we got 3 sequences. Okay, are you able to see all the 3 sequences? And we marked some of the residues in red. If you look at these 3 sequences and the marked residues, can we tell something from any specific characteristics of these sequences? You can see some specific characteristics. For example, if you see this one, right, you can see this region. Can you see this region? What is the characteristic of residues in this region? They are in FASTA format actually, because this is the FASTA format. It starts with the greater than symbol and the sequence. But if you see the red letters here, the highlighted ones, these are highly hydrophobic, so this could be in a transmembrane segment. In second sequence, you can see many charge residues, positive charge residues, lysine arginine. So it could be probably DNA binding domain. And the third one is lot of serines here. So serine is very important for the case of beta barrel membrane protein. So that is the reason I put like this, right? Fine. So now I give 2 examples. Okay, okay, I consider 2 different types of proteins. One is a globular protein, one is a membrane protein. So here I calculate the composition, right? How to calculate the composition? How to get the composition? What is the meaning of amino acid composition? Just now we discussed. Right, so you can divide the occurrence normalized with the n, right? Fine. So now I show the example. Okay, we collect a lot of data. From all the data, we derive these numbers, and I show the numbers here. So this is for the globular protein, this is for the membrane protein. So if you see these numbers, you can see some differences, right? Can you see some cases where they are similar and some residues are totally different, right? So if you see this one, it is almost similar, 5.97, 5.91, right? So it's both the globular protein or the membrane protein, both are same. But some cases, for example, you serine, this is very less, but here it is very high. This one is high here, but less here. This one is high here and less here, right? But the case of glutamine is low here and high here. So I show the figure because these numbers difficult to see for the biology. So if you see this one, easily you can see that some of them are very high here, some of them are very high here. And if you, I grouped the amino acids in various groups, aliphatic or aromatic or sulfur or polar or charged. And the polar ones, if you see, it is very high in the beta-barrel proteins, in the membrane proteins. And the charged one is very high here, the sulfur one is high here. Some of them has no change, there is no difference. Right? So these residues are the characteristic features of the proteins, and easily you can discriminate this type of proteins. Right? I'll tell you how to do it. Okay, so now I have one protein X. I don't know whether this protein belongs to group A or group B. The question is, can you exactly tell whether this protein belongs to group A or group B? Okay, simple algorithm I tell you. First you collect the data of the group A. For example, 100,000 of proteins from group A and 1,000 proteins from group B. Right? Because we got the uniprot. So where shall we get the sequence information? Which database contains protein sequence information? Uniprot, right? So uniprot has the annotation. So from the annotation, first you get the data for the take thousands of proteins from group A and thousands of proteins from group B. Fine. So you can calculate the composition of this A and the composition of group B. Right? Because you take the occurrence, normalize for the length, and you get these numbers. Right? Then we get the protein X. This we don't know whether it belongs to group A or group B. Get the composition. So we got 20 numbers. So right now we have composition A, we have 20 numbers, and composition of B, we have 20 numbers, and composition of S, we have 20 numbers. Right? So now relate these 20 and these 20. Compare this A, composition of A, and the composition of X. How can we compare this? Whether this is close or not? What are the parameters? For mathematics, you can easily tell what are the parameters you can use to compare these 20 numbers in A and 20 numbers in B, whether they are close or it is far. Can you tell one or a few features? Oh, deviation. It's very simple. That's good. So you can do the deviation or you can do the correlation. Because if the correlation is very high, then you can see that both have the same pattern. Or the deviation, if it is very less, you can tell that the numbers are very close. Right? Very good. Fine. So first you calculate the composition of A and composition of X and take the deviation because we take the absolute. Why do you want to take the absolute deviation? If you don't take the absolute, what will happen? Yeah, plus and minus will cancel out. Finally, we get close to 0, right? So either you take the absolute, or you can do the deviation. You can take the square root of the square summation of square root. So you can do that. Fine. So you take the difference. That is sigma A, and then the same X we compare with B. That is sigma B, right? So now we compare the total sigma of A and sigma of B, right? If the sigma of B is less, right, then the protein belongs to which group? B, right? If the deviation is less for A, and then it belongs to right? So you can do the math here, right? So just you calculate the composition, simple mathematics, right? And for the get the two groups and take a new protein, right? You either compare with A or compare with B. In equal case, but usually it's very, very rare. You don't get the equal case. In this case, you cannot correctly predict. You cannot distinguish using this method. Then you go with the correlation. Right? If it is exactly the same, you use the correlation, or you can use different other methods. Right? You can use it, but that is very rare. We don't get like that. Then another case you can use some error bars. They can use some error bars. So you can nullify that effect rather than exactly the this one. Right? You can see some errors and you can also get that one. Right? You can, there are various ways to do that. Fine. It's very rare case, but there is, I cannot say it's impossible, but that is possible. There are some cases, yes, you will get the equal case. In this case, we cannot discriminate using these features, right? We have to use other features. For example, like I discussed about hydrophobicity. So you, instead of composition, you calculate the hydrophobicity. Here, take the hydrophobicity here, and then you can compare, and then you can assign, or you can use the polarity. So you have various properties, and use any of the properties, right? You can discriminate, or you can combine. For example, this property should be more, and that property should be less, and with both you have the relationship. Then you can discriminate, right? Fine. So they can use that. That's a very good question. Yeah. So this is clear now? Fine. Now I show an example. Okay, here there's two proteins I gave. One is the DNA binding protein, one is the membrane protein, right? So here this is the number of residues, right? N, and this is the composition, and this is deviation. With the globular protein, this is deviation of membrane protein. And if you see here, right? So here this is less, 34, right? So then they classify this as a globular protein. On the right side, if you see, which one is small for the globular or the membrane? So what is the total deviation for the second case? One is 23, one is 16. Which one is lower? Membrane, right? Because this is membrane. And another case, if you look into this, okay, she asked the question exactly the equal case. So if you see here the deviation is very high. One is 16, one is 23. So in this case, with a confidence level that is very high, if it is very high, then you can highly confident that this protein is completely this is the membrane protein. But if you see here the difference is not very high. So depending upon the numbers, these absolute numbers, that also we can give you the reliability. How far the method is reliable. That is the reason we cannot predict 100% accurate. If you do like this, you can predict, you can discriminate up to the level of up to 80%. You can do that. Then we use other features, other parameters that I discussed earlier, and also some of them I'll discuss now, right? And we can improve the performance, right? That is the major aspects. Even if it is completely zero, you can use different features. In this case, you can use that features to predict this type of protein, right? Fine. Okay. And we have servers, right? So you can have the different servers here to discriminate. This is if you click here, and you'll give a sequence, right? And if you predict, you can here predict, right? Here, this is your occurrence, this is your composition, this is a deviation, and here you can see it's very, very close. 37.7 and 36.66. But we classify this as the beta barrel protein, membrane proteins, but the deviation is very less, right? Yeah. Fine. Okay, this is the applications of these features to discriminate. So I show another example. Right here we have the sequence. I have the sequence here. The question is, can you discriminate? This belongs to group A or group B. This is average value given for group A. This average value given for group B. I give the sequence. Now the question is, can we discriminate, or can we identify, or can you find whether this belongs to group A or group B? That is alpha or beta. What to do? Right. My question is to check the sequence which belongs to alpha or beta, A or B. So how do you do that? First, what you have to do? Okay, first you need to calculate the composition and get the deviation from A and the deviation from B, and then check the deviation and compare the deviations. You can predict whether it belongs to A or B, alpha or beta, right? So here this is 45 for alpha, 70 for beta, so the result is alpha. And second case, 36 for alpha, 18 for beta, so it is a beta, right? This is what we got it. Right? This is another concept. There's a paired preference. There we use only one. Here we use two. That means There we use how many times A, how many times C, how many times D, how many times E. But now, even if the A, but what is the neighboring one? A is with the C, or A is with the D, or A is with the F, whatever. Right? So here the dipeptide composition you can calculate in IJ, where I is the first residue and J is the second residue. Right? J multiply 100 for normalize. So divided by total number of times this residue is occurred. Right? I plus J, Ni plus Nj, or you can do it probability. If you want to get the probability, you can research the number of probable times Ni into Nj. Right? You can use any of these equations to calculate dipeptide. For example, if I give this, right, this is the sequence, right? What is the dipeptide of PE? How many times PE occurs? Two, right? So how many times, how many P and how many E? How many P? 5. How many E? 5. So then this will be 20, right? 200 divided by 10, that's equal to 20, right? So can you calculate where the residue pair preference for PE? Or you can put EP, okay? Can you calculate this for EP? It's not the P, this you can calculate this. Can you calculate the pair preference for EP? Right, 400 by, how you get 20? How many E's and how many P's? 1, 2, 3, 4, 5, 6, 7, 8. How many E and how many P? Total EPs are 4, right? EP1 is here. 2, 3, 4. That's correct. 5 E and how many P? Okay, then the answer is 4 multiplied 100 divided by. What is my question I gave? EP, okay. Right, 10. This is not 20, right? Fine. Okay. So now we got these results. So you can do it. So here this is a sequence. I showed the same sequence earlier. So if you take the AA, this is the number. Fine. Okay. So now what are various aspects we discussed so far? Correct. That's what is correct. So what are various aspects we discussed so far? Composition. Before that, occurrence. Yes. Molecular weight. Yes. Yes. Hydrophobicity. The properties. Yes. And then residue pair preference, right? So then we discussed about one of the applications, so how to do it, right? So I'll show you a little bit more. It has a little bit some more applications, right? Fine. So here now I have this sequence here, right? And I gave these numbers. Already I gave these numbers earlier. The same number I use here. So you can make a profile. Right? You can make a profile, right? Using this one. What is a profile? Profiles will tell you, the hydrophobicity profiles will tell you simply plot of the hydrophobicity indices, of the residues against the sequence numbers. That means x-axis you can have the sequence, on the y-axis you will get the numbers, right? For example, if you have a pen and paper, you can take a pen and paper, you can draw a graph, right? And x-axis you write at least some of these sequences, the residues. And the y-axis you plot. For example, S, the value of S is -1, so you make a dot, and A is +1, M is +1, P is -1, and you get these numbers, and you will get a such of lines, and that plot is called header. I'll show you very soon. I'll show you how to get that. Right? That plot is a heterogeneity profile which connects the amino acid sequence versus the heterogeneity indices. Okay, I told you that this is the numbers, but that's not random, right? But these are numbers I put for the properties. But here I give the exact numbers. Okay, here these are the exact numbers. Okay, here this is the values proposed by Nozaki, Tanford, Jones, right? So here you can see the numbers. Can you see whether the tryptophan and the phenylalanine, the values, right? You see here, tryptophan and phenylalanine, I get very high number, right? And the isolation. Can you see the numbers here? Tryptophan, phenylalanine, and isolation, F, W, and I. The values are very high or very low, comparatively. What is the value for I and what is the value for W? A occurrence I started in the beginning. That is just a count. How many times A appears, how many times D appears, how many times E appears. Just a count. That is the occurrence. Okay. So now what is the value for I? What is the value for W? Very high, right? If you see the right side, this is the experimental scale. This left side is experimental scale by the Nozaki, Tanford, Jones using the ethanol water experiments. This is the method, the scale developed by us, right, in our group. So here also we calculate the values. These are computational values, right, depending upon the occurrence of residues, right, which are close to each residue. Can you see here what is value of W and what is value of I? Okay, that's very high. So that means you can see the coherence. So here also you can see W and I is very high, and here also you can see W and I are very low. Here also W and I are very low. Then go with this one. Go with the -2. For example, you take the RRE, right? So you can see here lies like K RE or RRE. What is the value for R? RK, if you say the K, lysine is what is the value of lysine here? What is the value of E? What is the value of K? (1.64K). What is the value of E? 0.67. Here, what is the value of K and what is the value of E? Right side. Okay, see here also it is very less compared with the other residues. Here also it is less, and here also if you see the K and S, they are very less, very highly polar, right? So you can see the correspondence. So if you take many scales, currently around 300 to 400 scales are available in the literature, right? These are heterogeneity indices. This is the heterogeneity indices for 20 amino acid residues obtained from experiments, and this is the one GM. This is the heterogeneity scale. That means the indices for the 20 residues obtained by the three-dimensional structure analysis. Right. This is the experiment. This is by computation. Right. So here you can see the almost all the residues. The qualitatively you can see they have similar behavior. There are few residues which are different. That's mainly because only the proline is here. Here, because of this amino acid, it is having the high values here in the experiments, but here it is surrounded with the polar residues. That is the reason why it is less here in this case, right? So these are the hydrophobicity indices for the 20 different amino acid residues. Currently, we got 300 to 400 different scales available in the literature, and we can use any of these scales. Fine. So now we can use this one here, and here this is your profile. Okay, so you're asking whether you need to smooth it or not. So if you without smoothing, so right, we can see this graph. Right. So x-axis is amino acid residue, and the y-axis is the hydrophobicity, right? And each value you take from the for example, you take first residue and take the value from here, right, and then plot it. Right. I'll ask you to plot maybe very soon. Right. So then we will get it this graph. This is without smoothing. That is the reason we get the very random. They can see the plots. And the second aspect, we can also use the plot. We can smooth. Right. That means if you take the window smoothing of 9 residues, that means take the average of these 9 residues, or take the average of 9 residues left side and 9 residues right side, and take the average. If you do that, you get the graph like this. Can you tell something from this graph now? Right, you can see some very peak here, this side, here, right? These are the peaks. That means these regions are highly hydrophobic. These residues are highly hydrophobic, and actually these are the transmembrane helical segments. And this protein, 1-PRC, this is a photosynthetic reaction center. This is the first membrane protein solved in the literature, and the researchers who solved this structure got the Nobel Prize, right, in 1989. Fine. So here this is the one. The same sequence I use here, right? But here we couldn't find, because you can see it here, but that is not so clear. So when you smooth it, you can easily see it. So that means this hydrophobicity scale, you can use it to predict or to identify the membrane spanning regions, right, in any of the transmembrane proteins. If you give the protein sequence, just you plot it, right, and we smooth it, and easily you can see, okay, this is region 1, this is region 2, region 3, region 4, region 5. It has 5 transmembrane segments. Easily you can see all the 5 regions from this plot. Very simple diagram, right? Fine. Now this is another one. This I will ask you to plot now. So can you plot this sequence, left side sequence and the right side sequence, and give you give your interpretation? You can show in your video if you want. Use these numbers, right? Average value, take it as 0, and make a plot for the sequences given the left side and right side. How to draw? Take the first this axis, you put A, I, L, B, Y, and A is 1, I is 2. So make these numbers and make the plot. Likewise, for second sequence, you make a plot like that. No, not adjacent effect. You just plot exactly the same. A is 1, and I you can take 2. Here L you take 2, B you take 2, like that. If you take this one, A is 1, K is -2, like that. So now put the central average line again. It's required to put the average line. Average you take it as 0. If anybody drew the plot, just let me know. I will move on. You can use your video to show the pictures. Others can also see that. No, you don't have to use Excel and all because there is just 10. I mean, if it is sequence very long, then you do it off. Even 10 days is fine. You can take a pen and paper, I can draw. This is what I usually ask my students to draw on the board. Yeah, can you show the picture so that others can also see the picture and they can verify whether what they have done is correct or not. I want to see the pattern. Okay, no issues, fine.
Sir?
Yeah, fine.
Sir, sorry for interruption.
Yeah.
Sir, the expert talk we can wind up here, sir?
No, under five more minutes I'll finish it up, yeah. Okay.
Okay, sir. Okay.
Yeah, no problem. Okay, I'll finish in 5 minutes. Okay. So you draw this, so you will get the pattern like this. Okay. Here I showed the pattern. You will get the pattern like this. So here it is highly hydrophobic. This is the part of transmembrane segments, and here you can see one is high and one is low. You can see the periodicity of one low and high. This is mainly because of the beta strands. That will explain tomorrow the secondary structure, right? So if you get this pattern, you can easily predict this segment belongs to beta strand. If you get this type of patterns, you can easily say, okay, this belongs to the transmembrane helical segments. Okay, so you can do it later, and then you can write out the sequence, and you can draw later, and then see that you will get this pattern. Okay, so now exactly we have the pattern definition. Maybe another 5 to 10 minutes I'll finish it up. Okay, so here if you see, this is the definition for a pattern. So there is a square bracket. So square bracket means any of the residues within the bracket is allowed. That means L, I, V, M. So any of these residues is allowed. And here also you can the possibilities are VIC, anyone is allowed. X2 means X is any amino acid. You can put any of the 20 amino acids 2 times, right? If they write any specific amino acid G, that means that place is occupied only with the G, right? You cannot change this amino acid. It is very conserved that you have to keep. Here one among these, and here also one among these, X is any 2, like that, right? If there is a curly bracket, then you have to exclude. If there is a curly bracket and if you put A, then A is not allowed at that particular position, right? So if you take this one, then how many positions in this sequence? This is 1 position, this is 1, 2, here 2, 4, this is 1, 5, 1, 6, 2 means 2, 4 means 4 times. How many positions in this sequence? Can you tell quickly? There are 17 positions, right? So you can 17 positions, and can you form your pattern? Can you form a sequence for this? Okay, so I'll tell you this one. Okay, here this is your pattern, and there are 17 residues, right? So this any of 1 will occupy at position number 1. This any of 1 will occupy at position 2, and any residue in positions 3 and 4, and G at position 5, and so on. Right? Okay. Now I give you a simple pattern. Right? Can you give me one pattern for this one sequence? How many positions in this pattern? Six, right? So can you write the sequence for 6 residues peptide using this pattern? Correct. So can you write a peptide? So what are the residues allowed in the first position? A is correct, G is correct, and KK correct, and the X is M, fine. Now the last one, what is the meaning of this curly bracket? Curly bracket means you need to exclude. If there is C, F means these should not appear in that particular position. First, can you refine your peptide then? First 5 are correct. Okay, correct, fine. Right? So here the first one is anything within these 3. So you put A, fine. G, it is conserved, you cannot change. So G is there. K and R, any one among these K and R 2 times. So you put KK, that's fine. You put KK, KR, RR, anything is fine. And X is any amino acid. So you chose M, that is okay. Here WF5, exclude these 3 and put anything else. So you put A. Okay, that is fine. So anyone can write any, okay, that is fine. A, G, K, K, M, T. Okay, fine, right? So this is fine. Then, okay, you can write the pattern. This is the standard notation. You can use it. Okay, now if you use it in the PIR, you can search analysis. Go to the pattern search. Right? So here you write your pattern, and if you submit, and here you get all the information. Right? So here you can see this is your region where you can find the pattern. Right? Okay, so we did and this motif like this. We defined this motif. This is very important for the insertion of the beta barrel membrane proteins, right? And we published this in Cell, right? Because it is one of the prestigious journals because impact factor of more than 40, right? So we published in Cell because we identified this specific motif, right, for the insertion, right, and the assembly of these membranes in the beta barrel membrane proteins. Okay, I want to write an algorithm, but now we did because no time. This is fine. So this is the last one I show you in 2 minutes. Okay, there is the specific peptides you can search. Right, this is a pattern. Any specific peptides you can search. Go to this peptide here, and then here this is your peptide, and you go here, and you can see all the peptides here, right? And okay, I think I'll stop here. Okay. Okay, if you have any questions, just your time to discuss.
Yeah, sir. Thank you, sir. Thank you for your interesting and interactive session. Now, I request the participants, if you have any question, you can write it in the chat box, or you can unmute yourself and
Okay,
ask
I look
the
at
question.
the graph. Can you show the graph? I want to see the graph.
Yeah.
Yeah, because that will be good. Very good. Can you rotate? Because you need to rotate it. Okay. Okay. I could see it. Yeah. This is the first one, right? Yeah. It's good for other participants. Yeah. You can see all the residues are above the average value, right? So you can see they are highly hydrophobic. Yeah. And the second one, you can see the pattern. Second one, you can see up and low patterns.
I didn't draw, sir. I will draw.
Okay, that is fine. Yeah. Second one, if you see, you can see the patterns of low and high patterns. That is the characteristic features of these beta sheets. Okay, so
Yes.
yeah, because that's good for other participants if you see the graph. I showed in my laptop, but it's good. That's correct. Fine. So what are various aspects we discussed so far today? So can you remember, can you tell what are the various aspects we discussed today? Okay, before composition we started with structure. What is primary structure? Okay. Good. It's good you remember many things. Okay, fine. Also, I get the response from different participants. That is also interesting. Okay, how to get the molecular weight? This is the composition, correct? And the molecular weight, you need to minus 18 and minus, that's fine, right? Okay, then what else we discussed? How to discriminate the two classes of proteins based on amino acid composition. We can do the same with the pair preference also. Okay, then we discussed about the patterns, right? And the peptide match, right? Fine. So it's good, at least I remember what are the aspects we discussed today. Okay. Well, actually, I prepared a lot, but I don't have time to do all these things. Okay, so if you have any questions, just we'll have the questions, or just we'll wind up. We'll see tomorrow about the secondary structures. Yeah. Or I'll continue this tomorrow and then go with the secondary. It depends. My slides are available in my this one. What's that? NPTEL. Check the NPTEL. Their better qualities are available and more details. This is why I encourage all the students to take notes. Usually I don't give it my slides. I ask them to take notes.
Yeah. Any more questions from the participants? Okay. Okay. Then.
Okay. Yeah.
We can once again thank Professor Michael Gromia sir, and I also welcome Dr. Berlin sir, who was chairing this session. Yeah, so Dr. Berlin sir actually was a research scholar at IIT Madras. Basically, he hails from Marthandam, Tamil Nadu, and he is presently working at Civil Engineering Department, and he is Associate Dean for Planning and Development, along with TechCube Coordinator. So he was chairing that session.
Sir.
Yes, sir.
Yes, sir. Actually, sorry, I couldn't join in the beginning due to some network issues. And thank you so much, sir, for joining with us in this today's talk. Professor Gromia, sir, I think today's talk is, I hope, very nice for the participants. I couldn't hear full video anyway, full lecture. Anyway, thank you so much, sir, for joining with us and giving this opportunity to the participants for sharing your knowledge with us. Thank you so much. And once again, I thank our coordinator, Dr. Vanakumar sir, and our his team also. Thank you so much, and all the participants also. Without them, the workshop may not be a fruitful one. So thank you so much for joining all the participants. Thank you so much, sir. I think Vanakumar, you can continue, please.
Okay. Okay. So one of you asked about the motive. Motive, maybe we will discuss tomorrow more details. Motive will tell you any specific residues. For example, 5 residues, 3 residues, 4 residues. Any specific residue. For example, I told the beta valve membrane proteins. I have the polar, non-polar, polar, non-polar, G, polar, non-polar pattern. So that is specific motif. Motif means you can tell the specific residues which are located at specific regions, right? So there is a tripeptide or tetrapeptide or pentapeptide. Sometimes some residues can give a gap. So for example, if you, I can. Just will not
Sir, is it over?
Just 1 minute. Yes.
Okay. Sir.
Okay. So here, if you say this is a kind of motif, that means you can see it here. This is a segment of 1, 2, 3, 4, 5, 6, 7, 8 residues. 1, 2, 3, 4, 5, 6, 7, 8 residues. And here G is occupied at the particular position. There is only G. And PO means polar, and HA means hydrophobic, small hydrophobic, and capital HO is this one. So any number of residues, mainly it is less than about 10 residues, right? You can have a specific, either all the residues are same, right? For example, AKI, that you can do it, or you can with some X, or you can with some possibility of combinations. This is the specific segments, and we call this as a motif. So this is called a beta signal motif. We call it as, because this is having the beta barrel, beta strands, and this is important for the assembly of these proteins, and this is available, right? You can see the occurrence of this in almost all the mitochondrial beta barrel membrane proteins, right? I have the data, right? So that is the reason why we call it beta signal motif. Yeah. If you have more questions, we will discuss tomorrow.
Yeah. Okay, sir. Thanks. Thank you once again on behalf of our director, institute, and participants, on behalf of our chair and myself. So thank you, sir.
Okay.
Thank you, sir.
See you tomorrow. Yeah. Tomorrow is 11 o'clock, right?
Yeah.
Okay, I'll continue and then I go.
Yes, sir. I need to check.
Yeah. Tomorrow is. When is?
I think maybe first 9 to 10:30.
Oh, tomorrow is 9 to 10:30. Okay. I thought it is 11 again.
Yeah, no.
No, it's okay. 9 to 10:30 is fine.
I will recheck it.
No, it's okay. 9 to 10:30 only, because I have.
Yeah, tomorrow 9 to 10:30.
I have class at 11. Yeah. Okay. Yeah. Okay, I'll do it tomorrow morning 9. So
okay, sir.
Okay, then yeah, fine. Okay, bye.
Yeah. Thank you, sir. So participants, this is for the first half. Next half will be starting at 1:45. We will continuing with Professor Parimal sir. So join us 10 minutes early. Okay. So I'd be happy if all of you are on time. Okay. Yeah. Thank you for your cooperation.
Yeah, Banav sir.
Thank you.
Do you have? Yeah, Banav sir.
Yes, sir.
Yes. Do you have any class at 3:30 today?
Yeah. Thanks for reminding. So we have worked out on session 3:30, starting 3:30. So you are requested to join that too.
Okay. All right.
So I'll just post that. Yeah.
Okay. All right. Thank you.